Hi, this is Ajithya. In this video, we will discuss about parasitic protozoans. Parasitic protozoans live in the body of other organisms, and these organisms are called hosts, and the parasite depends on this host for food, shelter, and other requirements. Parasitic forms are present in many phyla of Protists. However, some phyla such as Rhizopoda, Kinetoplasta, Epicomplexa, Mycosporidia include large number of parasitic protozoans. Kinetoplasta, Epicomplexa and Mycosporidia include parasitic forms only. The parasitic mode of life among protists has evolved as a secondary adaptation, and the parasitic forms have arisen from free-living ancestors. The host-parasite interaction is a negative interaction between two species in which one organism, that is the host, is harmed, whereas the other, the parasite, is benefited. However, generally there exists a delicate balance between the parasite and the host so that the relationship does not result in total destruction of the host. Now we will see the phylum Rhizopoda in detail. The term Rhizopoda means root-like feet. The phylum consists of amoeboid protists, example amoeba and similar unicellular organisms. This group was formerly called Sarcodina, and the members of this phylum are mostly free-living aquatic forms, but a few are parasitic. These organisms does not have a definite shape, and they move by means of a false feet called pseudopodia, which is a temporary extension of the cell. Pseudopodia is also used for feeding by engulfing the prey and incorporating it into the cytoplasm. Sometimes the pseudopodia may be finger-like blunt projections called lobopodia or thin filamentous philopodia. The food remains inside a vacuole called the food vacuole, within which food gets digested and absorbed by the cytoplasm. The undigested food is ejected through temporary openings. In these organisms, osmoregulation is effected by specialized structures called contractile vacuoles, which contract intermittently to release the excess water to the surrounding medium. Amoeba reproduces asexually by binary fission or multiple fission. The common examples are Amoeba proteus and Entamoeba histolytica. In this video, we will discuss about Entamoeba histolytica in detail. Entamoeba histolytica is an endoparasite that belongs to the phylum Rhizopoda. It lives in the upper part of the large intestine of man and in the mammals such as dogs, cats, rats, and so on. Though Entamoeba histolytica enjoys a cosmopolitan distribution, it is more prevalent in tropical regions. The term Entamoeba comes from the Greek word ento, means from within, and amebe, which means change. In the large intestine, it lives in the mucus and submucous layers and causes a fatal disease called amoebic dysentery. It is called Entamoeba histolytica because it can cause histolysis in the tissues where it thrives. Entamoeba histolytica is a small amoeba ranging from 15 to 25 microns in length, and it is an endoparasite. It exists in two morphological forms, the trophozoite and precystic form. The active motile form is called the trophozoite, and it resembles amoeba in the structural details. The body of trophozoite is enveloped by a very thin, flexible, transparent, semi-permeable membrane called the plasma lemma. The cytoplasm is divided into an outer ectoplasm and an inner endoplasm. The ectoplasm forms a thin, granular, clear, somewhat transparent and firm outer layer, and the endoplasm is granular and more fluid in nature. Entamoeba histolytica has a single pseudopodia which is blunt and is called the lobopodium. The parasite hence is referred as a monopodial parasite. The organism undertakes slow forward movement with the help of this lobopodium. The endoplasm contains a vesicular and rounded nucleus, and it is bounded by a delicate nuclear membrane whose inner surface is encrusted with fine peripheral layers of granules called the chromatin granules. At the center of the nucleus is a very small endosome or a karyosome, which is dot-like. Other cell inclusions of the endoplasm are food vacuoles that often contain ingested erythrocytes and fragmented epithelial cells. And being a tissue parasite, Entamoeba endoplasm does not possess contractile vacuoles. The mode of nutrition in Entamoeba histolytica is a holozoic mode, and it feeds on blood corpuscles and fragments of epithelial cells and a few bacteria as well. This endoparasite is believed to secrete protein digesting enzyme that dissolves epithelial mucous membranes. It then ingests blood corpuscles with its pseudopodium. It can also absorb substances saprozoically from the surrounding medium. Now we will see how Entamoeba histolytica reproduce. Entamoeba histolytica completes its life cycle in a single host, and hence it is called a monogenetic parasite. They exhibit only a sexual mode of reproduction, and in the trophozoite stage it multiplies by binary fission. The trophozoite stage multiplies asexually within the intestinal walls of the host, and the nucleus divides mitotically into two. This is followed by the division of the cytoplasm by a constriction. The daughter cells then grow in size by feeding on the host tissue. They infect the host cells and multiplies again by binary fission. This cycle is repeated. Under certain conditions, some daughter cells fails to grow and escape into the lumen of the intestine. These small rounded forms remain sluggish and are called precystic forms or minuta. These precystic forms undergo encystment in the lumen of the intestine. They secrete a thin, flexible, retractile and resistant cyst wall around themselves. The cyst of Entamoeba histolytica is a spherical body measuring about 15 microns in diameter, and its cytoplasm is clear and hyaline and consists of two chromatoid bodies and many glycogen vacuoles. The chromatoid bodies are composed of ribonucleoproteins, and at this stage the cyst is uninucleated. Soon, this single nucleus divides mitotically into two and the cyst becomes binucleate. Next division results in 4 nuclei and the cyst now becomes tetranucleate or quadrinucleate. Now the cyst comes out of the body of the host along with the feces. The cyst can remain alive for many weeks outside the body of the host till it reaches a new host. The infection of the fresh host is effected by swallowing the cyst along with contaminated food or water. This method of infection is said to be a contaminative infection. Houseflies, cockroach and other insects may also carry the cyst from the stool of an infected man and contaminate the food and water. After that, excistation takes place in the intestine of the new host. Here, the cell wall is digested by intestinal enzymes. The tetranucleate parasite is set free into the intestinal lumen, and this stage is called the metacystic stage. Excistation is simply the process of transformation of the cyst into trophozoite stage. Now, the metacystic form undergoes a series of nuclear and cytoplasmic divisions producing uninucleate daughter amebulae, and the young amebulae become actively motile and invade mucus and submucous linings. These then develop into trophozoites by feeding and growth. It is in fact the tissue invading trophozoite cause the characteristic amoebic dysentery. The trophozoites penetrate deep into the mucosa and submucosa of the large intestine and will cause necrosis. The penetration into the host tissues is achieved by histolysis, and hence the name histolytica. Within the intestinal tissues, they multiply by binary fission and spread radially outwards to create flask-shaped bleeding ulcers. The cavity of ulcers are generally filled with mucous bacteria, parasites and cell debris. The ulcers pour their contents into the lumen of the intestine, and these are discharged out through feces. In amoebic dysentery, the stool is usually acidic and the stool contains blood and mucus in which swarms of amoebae are usually present. The patient shows signs of discomfort owing to the intense pain in the abdomen because of highly frequent passing of stool. Sometimes the trophozoites pass into the portal circulation and enters the liver. In the hepatic tissues, they settle and form abscesses. During this condition, the patient suffers from liver pain and develops fever. Abscesses may also occur in the lung during extreme cases. Through blood circulation, the trophozoites may also enter spleen, gonads and brain, where they can destroy the tissues. In severe cases, the infection might prove to be fatal. Some prophylactic or preventive measures against this Entamoeba histolytica are sanitary disposal of fecal matter, washing of hands with antiseptic soap and water before taking food and after using toilet, protection of food and water by contamination by house flies and cockroaches can also prevent the spread of Entamoeba histolytica. Using hot water for drinking is yet another method for preventing Entamoeba histolytica infections. There are other species of Entamoeba as well, like Entamoeba coli is one of the most common species found in approximately 50% of human population in the world. Entamoeba coli also lives in the lumen of the colon, but it does not enter into the tissue walls unlike Entamoeba histolytica. It is a non-pathogenic species as it does not cause any disease in man. There are other Entamoeba species like Entamoeba gingivalis, which is found in the mouth of human beings. Thank you for watching the video. Hit the like button if you loved the video and click the bell icon for notifications.